Greetings. Today we'll be discussing the paper Quantitative Interrogation of Protein Coaggregation using multicolor fluorogenic protein aggregation sensors by Liu, which was published in Chemical Science in 2021. Depicting the coaggregation process is essential to delineate the disease mechanism and also identify authentic drug target. Most of the protein aggregation detection methods are limited to a single protein investigation and hardly resolve the issue of coaggregation of different proteins. In this paper, the authors created a dual color thermal shift assay, TSA in short, that orthogonally and quantitatively monitors two different protein coaggregation processes. They took 4 environment-sensitive fluorophores with spectral coverage from 500 nanometers to 620 nanometers for their sensor. It was made sure that the spectral coverage of the sensors or the fluorogenic components was expanded in order to determine the protein aggregation of a wide range of proteins. The current assays use just one Cipro-orange probe to detect single protein aggregation, whereas in this work, they used orthogonal fluorescence readouts to monitor dual protein coaggregation simultaneously. Out of the 4 fluorophores they have chosen, CCVJ and Mero were the best suited to serve as the sensor pair. Though the insoluble protein aggregates retain fluorescence, fluorescence can also arise from early aggregation precursors like unfolded proteins or misfolded soluble oligomers. To rule this out, they used urea-mediated unfolding and no fluorescence was observed. They also used turbidity assays, which proved that the signals occurred at lower temperature than the OD333 signal. This suggested that fluorescence may be arising due to earlier formation of insoluble aggregates. So, unlike the other amyloid sensors that report a much late-stage aggregated species, these sensors were capable to detect early misfolded soluble oligomers. On the other hand, they also developed a system to examine the individual roles of TTR and APOA4 proteins, which when co-aggregated lead to cardiomyopathy. TTR was labeled with CCVJ and APOA4 was labeled with Mero. They found the presence of the aggregated APO4 protein can slightly stabilize the TTR mutant and inhibit its aggregation, suggesting its role as a pathological chaperone. These sensors thus provided quantitative analysis on the thermodynamic interplay during protein coaggregation. Future color enrichment of the sensor toolbox would help in detecting more and more complex disease model systems. Thank you.